Okay, hope you're having a good day. This is a pre-lab. First thing we'll look at is the lab on photosynthesis. And on this lab, we're actually doing two labs. One is on photosynthesis and the other one is on cellular respiration. And so we'll have to split this up, and half of the students will do some of one lab, and the other half will, of course, do the other lab and pieces of that one. So we can't all do all of it. It's just too much to actually get done. So, and that's okay. We'll discuss it before we leave, make sure everybody understands what did happen and what should have happened if something happened that shouldn't have happened, which happens occasionally. Okay, so on photosynthesis, you can see that we're taking carbon dioxide and water, and we're going to produce sugar, C6H12O6, plus we'll produce a little bit of oxygen, plus we'll also make a little bit of water. So that's your overall equation. It doesn't look like the one in the lab book, but this is the photosynthesis equation that you will need to know. Okay, one of the first experiments that you'll be doing, you're working with a process which is called chromatography. And on that process, what you'll find, you'll get this little jar. And on that jar, you'll have a stopper that'll have a paper clip in it, and then you'll be provided a slip of paper. This is a special kind of paper called chromatography paper. You're going to add some solution, but not much. It's called chromatography solution. And you also want to add a pigment. So the way to really do that pigment, the best way to do that is to take a piece of spinach and lay it at the bottom, about 1 centimeter at the bottom of this paper, and just roll the pair of scissors on top of that, and you'll see you'll get a green line on that, and that green line is your chromatography line, pigment line. And then, of course, what's going to happen, the chromatography solution, which is the red, is going to pull up this paper. And as it pulls up the paper, then what we're going to do is we're going to separate that pigment into different colors. So you'll separate it into maybe a couple of green colors. You'll get a couple of different green colors, so you'll have one and then a second and then a third and so forth. So you get some different greens, you get some different yellows that will separate and so forth. And so we'll have a couple of yellow lines, and you'll find one yellow line way at the top. Okay, so really we're separating the pigments that you find in spinach, and that whole process is referred to as a chromatography, just separating out those pigments. Okay, one thing we'll want to calculate is what's called the ratio factor, which is very simple to do. You just take the distance moved by the pigment. So our pigment, we have different pigments, as you can see here. We have the bright yellow one at the top. This will be a pale yellow, and then you have a couple of green ones. So you want to see how far from the original line, say like the first green one moved, and how far the second pigment moved. And that's the distance the pigments move, and then of course you have the third, and then of course the fourth pigment there. So distance moved by the pigment divided by distance moved by the solvent, and that's that solution that you put into the base of this container here. Make sure that that solution is below and just barely touching the bottom of that chromatography paper. You don't want to dissolve your pigment line, which is right here. You don't want to dissolve that into the solution. You want it to rise up and separate. And so what you're doing is giving a very definite number called a ratio factor, and that ratio factor is very distinctive for different pigments. That's how they know what pigments are actually in a particular plant. We just typically use spinach. We could have pulled a leaf off of a tree. We could have used any number of different pigments. Okay, so that's one experiment. The other experiment you're going to do, you're going to take a large test tube, and in that test tube you're going to place some Elodea, which is a water plant, and we have some back there in a small aquarium, and we want actually, let's say, 10 centimeters, just so that we'll make it consistent, of Elodea. A lot of people put this in their aquariums. They think it gives the aquarium more oxygen. In reality, it doesn't. It provides well, it provides a little bit of oxygen, but certainly not very much oxygen. Okay, and you're also going to want to put in some sodium hydroxide. Now that's an NaHCO3. That's really just baking soda, sodium hydroxide solution. It's not a very concentrated, about a 3% sodium hydroxide solution. And then you'll want to stopper this thing, and make sure the stopper's on tight, because we want to make sure that we trap any gases that are being produced. Well, the gas that should be produced by that plant is going to be oxygen. So the plant has the ability to extract CO2 out of the sodium bicarbonate. So really the sodium bicarbonate, this is a source of carbon dioxide. That's why we use it to begin with. It just provides more CO2 than what that water would have in it, because this is a solution of sodium hydroxide and water. If we just put water in there, there's not a lot of CO2 dissolved in that water. So what happens then, the more oxygen that's manufactured by this Elodea plant, it's going to create pressure, and then the water is going to go up into this tube, and it will move a certain distance along that tube. So you want to measure the distance that the water actually moves. So really, if you start with the fluid level somewhere in here when you begin, so make sure that it maybe begins there, and then after, we'll give it 30 minutes. After 30 minutes, then you'll find it's only going to move so far in 30 minutes. And just measure that distance. We'll measure it in, say, centimeters. And in 30 minutes, then what we can do is multiply that by 2, which of course would be per hour. So it will move, let's say it moves 7 centimeters per hour. So that will be our photosynthetic rate of photosynthesis. So we're just determining rate of photosynthesis. One thing I left out, this does need to have a light source, so we should be able to, there's some light bulbs that we can plug in, and so I'll try to draw a light bulb here that does give off, of course, light. So that just provides a little bit of light for that process. Okay, so that's someone else can do that within the lab. So we're just going to try to divide this up quite a bit. Like I said, there are a number of different things that we will be doing. Okay, and you will find a graph in your lab, and in that graph what you're going to do is you're going to see that it goes way up on purple, it goes up on blue, it dips way down at green, it goes up, and then it goes back up a little, and that will be red. So this is a purple pigment. So purple wavelength is absorbed to the greatest extent by plants. Blue is not absorbed as well. Green is barely absorbed at all. That's why plants are green. They tend to reflect that light. And red is absorbed actually second best. I don't know if I did that, but purple absorbed to the greatest extent, red second best, and blue wavelength of light would be absorbed third best, so not very well. And then, of course, green is hardly absorbed at all. It tends to be reflected out of the plant. So on this axis, we have actually absorption. And then on the other axis, we're looking at wavelength of light. And this is a little symbol that we use for wavelength, kind of an upside down Y. So that's the absorption spectrum. And the point in having that graph there is so that everyone can understand that purple light is the plant that plants absorb best and to the greatest extent. And green light is the light that they absorb least well. So that is certainly something a person would need to know about plants. And so that will also help us understand some of the other experiment that we'll be doing. Okay, so really not anything to do except look at that graph. And then the other thing we'll do is we will take a large test tube, and again we're going to put L-Adea in it. We use that quite a bit in photosynthesis. And then we'll put again a stopper with bent glass tubing like this. And again, we're adding a sodium bicarbonate, NaH, CO3. Once again, that's just a source of carbon dioxide. The plant can get the CO2 out of that. And make sure when you stopper it, the fluid, the sodium bicarbonate fluid is going to go up and pass the turn, so we have a start point. And then after 30 minutes, we should have somewhere down here a stop point, and then we can measure that distance in centimeters. All right. So, and again, so you've done this 30 minutes. You'll multiply it times 2, which would be an hour, and you measure that distance. Let's say it moves 8 centimeters per hour. That's a photosynthetic, again, a photosynthetic rate of photosynthesis. Okay, however, we're going to alter this a little bit. One person we can say would add, and all of these, let's say add 2 drops. So we'll add 2 drops of, let's use, for 1. Now your lab book doesn't actually tell you to do this, but I actually changed this lab up a little bit to make it work better. Red dye, which is just red food coloring, not a problem. And another person could use blue food coloring, 2 drops. Third person could use green food coloring, and again 2 drops. And then we want to find a combination that would give us a purple. Now, I'm not exactly sure what that combination is. Do the best you can. If you hold it up, say like to a light source, you should be able to, with a mixture of red and blue. So that's what we're actually mixing, red plus blue. Then you should get somewhere in the neighborhood of purple out of that. One more time, we should also, of course, have our light source here. That should be a bulb, and that will pass light through that, and you can measure. So one person will be using red dye in the sodium bicarbonate, one with blue dye in the sodium bicarbonate, one with green and one with purple. You basically have to turn that tube up and down in order to get that to mix well. So we're going to see and understand what these pigments are actually doing. If you add red dye to that tube, you're reflecting out the red light. If you add blue dye, you're reflecting out the blue light. If you add green dye, you're reflecting out the green dye. And if you add purple dye, you're reflecting out the purple light. Because all of those colors are coming from that bulb, the red, the blue, the green, and also the purple. Okay, so we're going to measure photosynthetic rates, see what happens with that particular lab. Okay, the other part of this, and we're doing actually 2 labs today, so then we'll have another person working on this one, and this is called Cellular Respiration is the name of the lab. It actually is something that accomplishes 2 things, not just cellular respiration, but there is another mechanism we'll look at called ethanol fermentation. Respiration, here we go. All right, okay. So on this one, and this one's actually pretty simple, what you're doing is you've got a flask, and in that flask you want to put some beans, and there are 3 types of beans that you can put in your flask, about all you need as far as number of beans. You should be able to find a 75 milliliter line, so about 75 mils of beans. And those beans can either be dry beans, so one person can use that. Another person can use soaked beans, so those have been soaking in water. And then the third set will be boiled. So I have boiled those beans, and I boiled those for at least about an hour and a half. So they were boiled for quite a period of time. All right, one more time. We're going to have a stopper here, but in this stopper we're going to have what they call a thistle tube. So it's going to look something like this, and it should also have a stopper in it. So we want to plug that off. And then you should have a second tube that's sort of bent glass tubing, and you'll put that in you'll put a test tube over it. And this will just have tap water. You don't need to use DI water. So this is just tap, something you get from the faucet, tap water. Okay. And then this thing actually sets, we'll let it set for best about 45 minutes. So again, this would be one person doing the dry beans, one person doing the soaked beans, one person doing the boiled beans. So once you get it set up, you're just waiting 45 minutes. Now, after the 45 minutes, you pour out the tap water. So tap water goes out, and what you replace it with, so into that, you're going to put a pH indicator, and this is called a phenol red. And it indicates an acid is its function. So, phenol red. Well, if your beans have produced any carbon dioxide, at the end of 45 minutes, you also take your stopper out, and then you take a beaker of just tap water. That doesn't look like a beaker. We'll make it look like a beaker. And you take a beaker of tap water, this looks more like a beaker, and so again, that's just plain old tap water. And you pour that tap water through the thistle tube, and then if there was CO2 produced, then of course what's going to happen, the CO2 is going to combine with the water, and you're going to form this substance, H2CO3, and that is an acid, that is carbonic acid. Now, of course, if you didn't make any CO2, you're not going to make the acid, and therefore nothing will happen with that phenol red color. But if you did make the CO2 and you pour it through the phenol red, because remember we've taken the tap water out after 45 minutes, we've replaced it with the phenol red, and then that's after 45 minutes. And then if you have CO2 in here when you pour the tap water through your thistle tube, you'll actually force the CO2 out and force it out, and then it will go over, and then it will bubble through that phenol red. And that's when the CO2, if you've made it, combines with the water, and you do make the acid. And if you do make the acid, the phenol red will then become a yellow color. So if you get a color change to a yellow, that means yes, I did make CO2. How do I know that? Because I did create the acid. All right, so that's one of the respirometer experiments that you do. So one person will use dry beans, one person soaked, one person boiled. So there is a lot to do here, but if we divide up the responsibilities, I think it actually works fairly well. Okay, the very last thing that we'll do, we're going to be looking at a process which is called a fermentation. Actually, 2 different types of fermentations, but This particular one that we're looking at is ethanol fermentation, which is how they produce alcohol. So how they make any kind of alcoholic beverage, whether it be beer, wine, liquor, all of that is produced through an alcoholic fermentation process, or ethanol fermentation. Okay, so this one does not require oxygen, as did our cellular respiration. We are going to begin with some sugar. We're going to vary our sugars. So C6H12O6, and then we're going to react. Notice I didn't add oxygen into that. We're going to allow the reaction to occur, and we're going to produce some carbon dioxide, CO2, and we're going to produce some ethanol, C2H5OH. And if you wanted to know the chemical formula for beer, wine, liquor, you're looking at it right there, C2H5OH. Plus, in addition to that, we make some energy in the entire process, which is not much, about 2 ATPs. Okay, so the way this thing is set up, and again, one person will do this part. You will take a test tube and you will add 2/3 of a sugar. Well, we actually have 3 different sugars that we'll use. So one person will set this up using glucose. Another person will set it up using fructose. A third person will set it up using sucrose, which is just regular table sugar. And then one person will set this up just using DI water, distilled water. And that is, of course, our control is what we're looking at here, just the control. All right, so we don't expect anything to happen with that water. So, and the other thing we're adding here is a yeast. So we add a yeast solution. Actually, it's really referred to as a suspension because it doesn't really dissolve. So I'll do that correctly. So 1/3 of the test tube length, and that will be the yeast suspension. Okay, so this is actually a small test tube. And then what you're going to do is you're going to invert this into a larger test tube. So we have 2 different sizes of tubes. So this one that you've set up with the sugar and the water, we're going to invert into a larger test tube. So, and I'll try to help you if you have problems with that. But let's say this is the larger test tube here. I'll label it large test tube. That's the way I usually put test tube. And then the small test tube would be inverted upside down. And this, of course, will be the one that has the yeast and the sugar. So yeast plus sugar. Or actually, we've got one tube that has yeast and water. That's a control. Nothing will happen with that. Okay, so if we get fermentation happening, what begins to occur, we begin to get some carbon dioxide collection within the top of this inner tube. Okay, so you can measure how much gas is produced. So if we produce more gas, then it has the ability to metabolize that sugar better. It could be glucose or fructose or sucrose. Or if it produces less gas, then it means it can't metabolize that sugar as well. Okay, now this also goes in a water bath. So there will be a water bath set up, and that should be at 37 degrees Celsius, which is actually our body temperature. So at the end of, let's let this thing run for 30 minutes. Then you go in and measure how much gas you've collected. All right, so as you can see, there's enough for everyone to do, but you're not doing all of it. You're just doing one specific aspect of an experiment, whether it be the glucose or the fructose or the sucrose. which I see I misspelled. It should have an R in it. But should be an R in here. There we go. Put an R in there. Anyway, so, or if you're using the beans, you're using dry or soaked or boiled. Different people doing that, or, you know, depending on what the experiment is, you know, you as an individual will just only be doing one aspect of that. Y'all just need to divide up the responsibilities, and we'll talk about our results at the end of this lab. And it tends to run fairly quickly. So as long as you've listened to the pre-lab and everything's cool with that, then you should be able to divide up responsibilities and run this lab, and it should have no problems. Okay, so good luck with the lab, and we'll see you on Thursday for that. Thanks.